AC चला है ना, fan off करिए, sound कम.
Yes, Darshan, you are audible.
Yes, sir. Yes. Hello, good morning to everyone. Welcome to today's webinar on topic what to look for: sustainable ways to protect the forest health, organized by Forest Protection Division, Tropical Forest Research Institute, Jabalpur. I am Dr. Darshan from Forest Pathology. To this webinar, I have some key points. This session is being recorded and also telecasted in Krushi Junction YouTube. Queries or questions during the sessions, please keep it in chat box, and guest or anyone will answer at end of each session. Now I am with permission of Director TFRI, I am starting this webinar. Now I am requesting our HOD Forest Protection Division, Dr. Nanita ma'am, to give welcoming words to our honorable guest and all participants. Yes,
Good morning, everyone. On behalf of Director TFRI, Dr. G. Rajeshwar Rao, I would like to welcome to all of you in today's webinar, especially Dr. A.S. Krishnamurthy, Professor and Registrar, Tamil Nadu Agriculture University, Coimbatore, and Dr. Anirban Roy, Principal Scientist of IARI, New Delhi. Welcome, sir, in this today's webinar. And before I start, I would like to brief about the TFRI, Tropical Forest Research Institute, is working for the forestry research, education, and extension for the Central Indian Zone. And we are also catering, from the Forest Protection Division, we are also conducting integrated insect pest management and disease of the forestry species as well as the medicinal plant species also. And we have some national repository of 750 butterfly species also, and we have developed trichocard for controlling the insect of teak, that is Eutectona maceralis, and and if you want any details, you can visit our website tfra.res.in. So this is from my side, and once again I would like to welcome to all of you for today's webinar. Thank you all. You can enjoy and listen our eminent speakers. Thank you all.
thank you, madam, for nice welcoming speech. Now we are grateful to and energetic, and also giving us opportunity to organize this event. Now I request to our director, sir, to enlighten the audience with his great words and inaugurate these webinar sessions. Welcome, sir.
Respected Dr. Krishnamurthy, Professor Plant Pathology and Registrar of Tamil Nadu Agricultural University, Coimbatore, and Dr. Anirban Roy, Principal Scientist, IR in New Delhi, and other outside guests, and my colleagues from TFRI. Good morning to you all. And the topic is very important in forest pathology. It's a really, the estimates say that entomological, insect pests and fires together are damaged by them together is less than the damage by forest pathogens. It's a main concern of how to protect the forest from these pathogens which is destroying the forest. You can see they compare the combined damage by insect pests and the fire are less than the damage done by forest pathogens. So it's a very important gray area where one has to work on it. The main problem in the forestry is that you cannot spray the chemicals as we are spraying in agriculture field. We cannot control the forest pathogen with spraying of chemicals. So we have to do some other ways. Only in nurseries, forest nurseries is possible, but not in regular forest. So we have to evolve some systems where we can control these, at least reduce the effect of pathogens on forest health. So the two guest lecturers who are Dr. Krishnamurthy, he will be dealing on mushroom cultivation, how we can best increase the livelihoods of the forest dwelling communities by way of growing mushrooms. And the second topic is on genome editing tool, principles and its applications in the management of insect pests and diseases, a very new area where we have to concentrate on how best we can use the genome editing tool in controlling forest pathogens. More so forest protection, but more so forest pathogens. That's the area. So I wish that the webinar planned by Forest Protection Division of my institute, Tropical Forest Research Institute, will throw light on important topics, these two important topics. I wish the seminar very successful. Thank you.
Thank you so much, sir. Now, I'd like to welcome our guest speaker of the session. The first speaker is Dr. to introduce about sir. And sir is currently working as a registrar at TNAU Coimbatore and completed his BSc Agriculture at ACRI Madurai and MS degree and PhD in plant pathology at TNAU Coimbatore. He is served as a assistant professor and later associate professor and professor in 2006 and 7. And he is also worked in Department of Forest Biology, Forest College and Research Institute, Metropolitan. He is also principal investigator, ACRP and Mushroom, Department of Plant Pathology, and he is also served as a professor and head Department of Plant Pathology and also a director, Center for Plant Protection Studies. And his significant achievements is introduction of commercial production technology for Milky Mushrooms, Alocipa indica. It is one of the first time in the world, and he was also responsible for the release of Oyster Mushroom and Pleurotus viruses APK1 1996, and also introduction of outdoor system for cultivation of Paddy Straw mushrooms as an intercrop with maize fields, and he organized several outright training program for cultivation of mushrooms and also organized every 5th of month in TNAU Coimbatore. And he's also served as so many, he's presented so many papers and more than 100 research papers in national and international recognized journals. And he is also editor of chief editor and president of Madras Agriculture Student Union. Like that list is so many things are going, and also sir has awarded more than 12 awards in academic and research achievements and visited Canada, China, Iran, Malaysia. I welcome you, sir. And our second speaker is one of the most young, dynamic, and great orator of our institute, that is Indian Agriculture Research Institute, presently working as a principal scientist in IRA, Division of Plant Pathology, that Division of Virology. He has completed his graduations in 1997 from BCKV, West Bengal. He also completed his MSc and PhD with specialization in plant virology from the Plant Pathology Division, IRA, New Delhi. Along with his academic career, he has maintained a very good record and received a gold medal in his MSc and PhD program. He also joined first as a scientist in Central Research Institute of Jute and Allied Fibers in 2003 in Barakpur. Later, he joined as a senior scientist in NBPGR, New Delhi, and he joined IRI in 2013. Presently, he is working as a principal scientist. His area of interest is in virus disease of the plants. He had identified several bigamo new virus new species. He is involving in teaching for the postgraduate studies, and beside this, his significant research is on for their management. And with this, I welcome you for this session. Thank you, sir. Today, our technical talk will start from the first lecture is given by Dr. Anirban Rai sir, and is talk on CRISPR, application of its genome editing tool, and applications in pest and management of crops. Please, sir.
Thank you. A very good morning to all of you. At the outset, I extend my thanks to the Director, Tropical Forest Research Institute, as well as the organizing secretary, Dr. Qasim, who happens to be our student at IRI. He was a brilliant student. So without wasting any time, today I'd like to discuss about the CRISPR-Cas genome editing tool its principle, its application in management of the insect pest and diseases. So I'd like to build this entire topic on different subtopics. First, I'll discuss about the genome editing technology platforms which are available. Then the scientific progress, particularly on the CRISPR-based technologies. Then landmark events and heroes of CRISPR, where I'd like to tell you about some of the developments, how this subject actually developed, how from a knowledge base, a technology developed, that history, how a PhD student's dream ultimately leads to the bagging of the Nobel Prize last year in chemistry. So these are all different kinds of landmark events, and who are the different scientists that associated with the development of this CRISPR technology. Then I'd like to talk about mechanism of the CRISPR-Cas9 as an adaptive immunity in bacteria, and how we are using it as a genome editing tool in the eukaryotic system. And then I'll talk about the application of the genome editing. First, I'll give an overview in agriculture, that what are the different aspects where we can apply genome editing. Then I'll focus on the genome editing, particularly for the biotic stress management, and where I'll talk a little bit about the forest pathology or forest plants genome editing, though very limited research work has been initiated in this field. And then particularly where is my strength, I am working on virus disease management using this genome editing. And there I'd like to mainly focus on a case study on chili leaf curl virus disease management using the CRISPR-Cas9. So this is a kind of roadmap that how I go one by one. So the genome editing, as the term indicates, it is a type of genetic engineering in which DNA is inserted, deleted, or replaced in the genome of any living organism using engineered nucleases, which we call them as molecular scissors. Now, genome editing is not a new concept. It started from long back in 1980s. And during that time, actually, the homology-based genome editing or recombination, where the swapping of the one gene with another gene we used to do in a modified cell. But the problem associated with this homology-based recombination is that identification of the modified cell is very difficult. It is just like searching a needle in a haystack. So genome editing was not progressed until and unless there was a very recent developments in different platforms using which actually the genome editing started developing as a complete subject. So during 1990s, the meganucleases were discovered. So this is the kind of nuclease which can cut DNA at a particular sequence. Then during 2000, the gene finger nucleases was discovered, where there are two components. One is detecting the DNA, and another enzyme can cut the DNA strand, double strands. Then during 2009, talent-based platform was discovered. And then in 2013, the latest addition is the CRISPR-Cas mediated genome editing. Now, if you see, except the meganuclease, the genome editing components or all these platforms has two components. One component recognizes specific DNA sequence, that is any specific gene or non-coding elements, and then a particular nuclease which can cut the double-stranded DNA and make a double-strand break. So all these three technologies are like that. But gene finger and talent, if you see, then for both gene finger and talent, we require a protein engineering for each cases, each selected targets, which is very much time consuming, resource intensive, and costly. Now, with the discovery of the CRISPR-Cas, it was appeared that this technology is much more simpler, they are more accurate and less costly. And that is why what has happened, if you see the scientific progress in the CRISPR technology is progressing much more faster than the zinc finger or the talent-based or meganuclease-based genome editing technology. So there is a phenomenal growth of the CRISPR publication in the short period from 2013 onwards when it was started. You can see the graph of the publication in the CRISPR-related genome editing is much more higher than the zinc finger or talent-based or meganuclease-based. So this clearly tells that the ease of doing of CRISPR-based genome editing and their accuracy is much more than the other technologies, and that is why CRISPR actually the technology of the millennium, because we are actually observing the very huge progress of this technology in terms of the publication and the output. So majorly, if you consider throughout the world, after the USA, then the China and the European countries and Japan, they are the main stakeholders for the research which is carried out throughout the world about the CRISPR-based genome editing. And there are several patents on one technology. You can see so many patents are there. And in the world, in history of the science, there is a biggest battle in the patent is going on between the two persons, actually two groups, to own who is the actually discoverer of this patents or this technology. Now, when we're talking about this patents and other things, definitely few people actually are getting the credits of patents, getting the credits of the Nobel Prize and other things. But any technology cannot be done by one or a group of people. So many scientists' small contribution actually are forming the base on which this CRISPR technology is now developed as a technology. So before going to start the in depth of the topic, first let me tell you about those small research work, starting from a PhD student to different other big scientists they have done to develop it as a technology. So in the last 20 years, total 9 countries actually involved in the development of this technology. The technology started during 1993 with the work of a PhD student. So he is that PhD student. Now he's old, but during 1993, his chairman was given him a project to identify the sequence variation in a salt-tolerant bacteria. So when searching of the sequence on those salt-tolerant bacteria, he identified multiple copies of near-perfect, roughly palindromic repeated sequence, which are separated by spacer, which is available in many bacterial genomes. And he observed that this spacer sequence has some sequence identity with some of the bacteriophages. In 2002, Janssen et al. Then termed those kind of near perfect, roughly palindromic repeated sequence, which is separated by spacer, as clustered, regularly interspaced palindromic repeats, which in short we call them as CRISPR. So this group also observed that, so this is the CRISPR loci where different repeat sequences are there, which is separated by some other sequences which we call as spacer. So ahead of this CRISPR loci, there are several genes, and those genes, they term them as CRISPR associated genes or Cas genes. Now these Cas genes also involved in this CRISPR and Cas mediated genome editing. Now, two gentlemen, Philip Hobart and Robert Bargo. Philip Hobart was involved in working in a company of production of the cheese and other milk products. So they are frequently encountered with the spoilage of those bacteria due to the attack of the bacteriophages. So Philip actually tried to find out that whether any technology is there using which we can get rid of those bacteriophages. So he come across with the literature available from Mojita and Jensen, and he and along with his co-worker, they first time showed that CRISPR is an adaptive immune system of the bacteria. So among those CRISPR and the Cas proteins, one of the Cas protein copy a portion of the invading bacteriophage and incorporated it as a spacer into the CRISPR loci. So whatever spacer is there, and the spacer sequence is actually copied from a bacteriophage when the bacteriophage infect a bacteria, and one particular Cas protein do this job. Then they observe that when a bacteria pauses a spacer sequence of a bacteriophage, then the bacteria is showing resistance against that bacteriophage. So this clearly tells that the CRISPR and Cas system is actually an adaptive immunity system of the bacteria. But they couldn't able to find out that what are the function of other Cas genes and how this actually, what is the mechanism of this adaptive immune system. Further, John van der Oost, he worked on this CRISPR mechanism, and he observed that from this CRISPR array, a long pre-CRISPR RNA is synthesized, and some other Cas protein, they come and they cut those pre-CRISPR RNA to form small CRISPR RNA. So this small CRISPR RNA, that time people did not know what is the role of this small CRISPR RNA in the adaptive immunity. Then Luciano Marafini in 2008 proved that the CRISPR RNA actually target DNA. But CRISPR RNA cannot cleave DNA, cannot degrade DNA, only they can target any DNA which is complementary to this CRISPR RNA sequence. Then Moinu in 2010 first showed that among different Cas protein, there is one Cas protein called Cas9, which is guided by this CRISPR RNA and creates a double-stranded DNA break. So CRISPR RNA along with the Cas9, they can cause a double-stranded DNA break. And they observed that the Cas9 protein cut the double-stranded DNA 3 nucleotide upstream of a NGG sequence and called this kind of sequence as protospacer adjacent motif. So when there is a protospacer adjacent motif, 3 nucleotides previous to that, the Cas9 protein can cut a double-stranded DNA, provided that the Cas protein binds with the cRNA and then target a DNA. But when he demonstrated that thing, he couldn't able to demonstrate in a in vitro system. That means if you take a DNA, a CRISPR RNA, and the Cas9 protein, it should cut in the in vitro condition. But he couldn't able to demonstrate that it is not happening. That means it indicated that something more is there along with the Cas9 and the CRISPR RNA. Some more components may be there. So that discovery came from a totally different lab, the lab of Emmanuelle Charpentier. So she was working on the next generation sequencing project of different bacteria, and she observed from that sequence that that upstream of the CRISPR and the Cas protein, there is another stretch of sequence, and very surprisingly, this sequence is actually complementary sequence of this repeat sequence. So whatever repeat sequences are there, direct repeat sequence, the same sequence in the opposite direction is present upstream of the Cas9 gene. And she also observed that until and unless tracer, they call them as the trans-activating CRISPR RNA, because this trans-activating CRISPR RNA helps the CRISPR RNA for their maturation and for the stability of the CRISPR-Cas9 complex. So she couldn't able to work further because she need a RNA biologist for that. So she joined hand with Jennifer Doudna. So Jennifer Doudna is a world-renowned structural biologist and RNA expert, and both of them, they then showed that two RNA, which is called the CRISPR RNA and the TRISR RNA, could function in vitro even when fused into a single guide RNA. So first they take TRISR RNA, CRISPR RNA, and the DNA to be targeted and the Cas protein, and observe that it can cut the DNA double-stranded, it can create a double-stranded DNA break. So when they join this CRISPR RNA and TRISR RNA together, they make a single RNA molecule, which they term them as single guided RNA, and this single guided RNA can also do the same function under in vitro. So now they have showed that the CRISPR-Cas9 system is actually composed of three components: a TRISR RNA, a cRNA, and the Cas9. TRISR RNA and cRNA can be joined together to make a guide RNA. So actually, we need two components, one guide RNA and one Cas9 protein, and that if they are available, then under in vitro condition they can touch the target DNA. So what happened? That after the discovery of the single guided RNA, this concept of the single guided RNA actually revolutionizes the CRISPR mediated genome editing, and that is why they were actually credited with the discovery of this CRISPR-Cas9 mediated genome editing, and last year both this lady got Nobel Prize in chemistry. But they couldn't able to show that really this kind of guide RNA can do the genome editing in vivo condition. So in vivo condition, genome editing was shown by Fan Zheng from the Broad Institute, MIT, and then he showed that under in vivo condition, he could able to create mutant genes with high efficiency and accuracy, and this paper actually initiated the era of CRISPR-Cas9 based genome editing. So starting from a PhD work of Mozita, ultimately the knowledge converted into a technology with the discovery of the guide RNA, and after its successful implementation in the editing of different genes. So there is a big battle going on between this Doudna and Feng Zhang regarding the patent of this CRISPR technology. Though Doudna and Charpenter got the Nobel Prize, Feng Zhang missed it, but this patent fight is still going on. We do not know who will win this patent. But this is the a small brief account on the discovery of the CRISPR as a technology. So from a knowledge base, how a technology developed, it is a very evident and very appealing aspect. Now, with this discovery of all this, I'd like to tell you what is the actual mechanism of CRISPR-Cas9 as an adaptive immunity in the bacteria, and how we are using it as a genome editing tool in eukaryotes. So what happened in bacteria, when this CRISPR array is there, Cas genes are there and TRISR RNA are there, then when a bacteriophage infects this bacteria, then it delivers its genome into the bacterial cell. And then this genome has many sequences, and one of the Cas protein, like Cas1 or Cas2, they targets this one portion of the phage DNA, recruit that and deposit it into the CRISPR array as a spacer sequence. So in that way, the process is called adaptation. So after infecting a phage, a portion of the phage genome is copied and incorporated into the bacterial genome as spacer sequence. So what will happen in the next stage? The CRISPR array produces pre-CRISPR RNA, and the TRISR RNA produces the pre-TRISR RNA, and then this pre-TRISR RNA and pre-CRISPR RNA, they, due to their complementarity of the repeat region, they form partial duplex structure, and with different enzymes, they then cut into different lengths of TRISR RNA and the CRISPR RNA, and ultimately a mature CRISPR RNA TRISR RNA hybrid is produced. When this phase is called the transcription and the maturation phase of the CRISPR RNA tracer RNA hybrid, and then when the same bacteriophage, if they infect the bacteria again, then already this kind of memory system is there, which recalls that mature CRISPR RNA and tracer RNA hybrid. The Cas gene, one of the Cas gene called the Cas9, got activated by binding with this CRISPR RNA and tracer RNA, and then this incoming bacteriophage, as they have the complementary sequence with this spacer, they will bind with this spacer region, and then this entire molecule is stabilized with the help of the tracer RNA, and ultimately the Cas9 complex become activated, and they can cause a double-stranded DNA break. And due to this double-stranded DNA break, there is a phage resistance. So this is the happening inside a bacteria, that how bacteria protect themselves from an incoming bacteriophage using this CRISPR Cas mediated approach. But how we are allowing, or we are applying this kind of immunity approach to a genome editing technology. So what happened? The CRISPR RNA and the tracer RNA, we can join them together to form a guide RNA. So if an inactive Cas9 is there, protein is there, we add the gRNA with that, then there is a conformational change of the Cas protein, which allow the binding of the gRNA with the protein. Then when a target DNA with the PAM motif is available, where a part of the target DNA is similar as the spacer sequence, then they will come and bind with the help of the PAM motif, they will bind with the Cas protein. And this guide RNA will now slowly unfold this DNA in a portion, and they will move on, and ultimately they form such kind of structure, and then 3 nucleotide upstream of this PAM motif, this HNH and RUVC domain, they creates a double-stranded break. And due to this double-stranded break, the DNA is getting clipped. But in case of eukaryotic system, we have a DNA repair mechanism. In case of prokaryotic system, we do not have, but in case of eukaryotic system, we have inbuilt DNA repair mechanism. Using that DNA repair mechanism, this cut end may be joined through non-homologous end joining, and during this non-homologous end joining, there could be insertion or deletion. As a result, there is a mutation in the genome, and in that way, we can create a new gene, we can stop the function of a gene, and we can actually modify a gene. On the other hand, if we provide, along with this system, if we provide a new sequence of the DNA, which is flanked with a portion of the cut DNA end, then this new DNA portion can join with this cut DNA through the homology directed repair, and in that way, a new DNA or edited DNA can be formed. So this is the way how we can apply eukaryotic system, this the bacterial adaptive immunity system in a eukaryotic system, and in that way we can create a new DNA, we can modify a DNA, we can stop the function of any DNA, and many things we can do using this CRISPR-Cas mediated genome editing. So in nutshell, the components of the CRISPR-Cas9 is this small 20 nucleotide spacer sequence. So this is the important things, that if anyone wants to initiate the work on the genome editing, they first have to identify a 20 nucleotide spacer sequence, which will be complementary with the DNA strand to which we want to create a cut. And this 20 nucleotide spacer sequence should have an NGG motif in the opposite strand. And then what will happen? This is called the spacer sequence, and this is the tracer RNA scaffold, which is universal, commercially available. This kind of universal sequence, and these two together form the sgRNA. And this is the protospacer adjacent motif, And in this Cas9 protein, there are two different domains. One is called HNH, which cut the complementary photospacer where spacer binds, and it is the photospacer which is cut by the RUVC in the target gene, which is this photospacer sequence is actually same. Actually, the power cut here, so suddenly internet gone. So anyway, so this is the system which is available for CRISPR-based genome editing. We have a Cas9 protein under a promoter, and there is a 20 base pair target sequence which you can clone under another promoter. And only for the genome editing work to be initiated, only we need to find out a 20 base pair target sequence which we can clone here. Then after production of the Cas9 protein and the guide RNA, they will join together to form the active Cas9, and we can now do the genome editing. So it is so easy now, very easy technology for editing the any gene or genome. So these are the basic steps actually which are involved in the CRISPR-based genome editing. The first step is the identification of off-target free, efficient, high GC content rich spacer sequence, which will be used as a CRISPR RNA. And finding out, for that, we need to find out 20 base pair region in the genome. It may be either host or pathogen, which is followed by a NGG sequence. Then we need to analyze those sequence for off-target effects through bioinformatics softwares. And then we need to check their effectiveness under in vitro condition, that whether those 20 base pair sequences can really cut a target DNA under in vitro condition. Then we need to develop the CRISPR-Cas9 construct through cloning, and that may take 10 days. Then delivery of the CRISPR-Cas9 module into the plant and the editing. So we may do with transient approach, that means delivery of the Cas9 and gRNA through binary vector, virus-based gRNA delivery into Cas9 expression plan, or we can do through the transgenic approach. And in transgenic approach also, we can keep the transgene as such, or we can remove the transgene, only we can keep the edited plants through selection. The latest technology is DNA-free delivery, where we do not use any construct for transforming the plant, only the protein, Cas9 protein and the gRNA directly bombarded into the callus. So genome editing occurs at the callus stage, and then suitable editing was happened. And then if the regeneration technology is very much available, then starting from the callus, we'll able to generate a edited plant. So there is no question of any transgenic. Then identification of the genome edited plant. After doing the genome editing, we need to see that whether genome editing has taken place and where it has taken place. For that, we have different technology like T7E1 approach or melting curve assay. And finally, we need to confirm the resistance through the challenge inoculation of the pathogen. And in that way, actually, we can proceed. This is the basic steps that how we can proceed through the genome editing. So when we have this kind of methodology and the tools are available, then the application of genome editing is very easy, and it is applied in invariably all the living organisms which are available in this world. Everywhere people are applying the genome editing technology for modifying the genome or genes. So particularly if you see on the agriculture aspect, started from different animal model, epigenetic variation, fuel, all those kind of things are there, but food aspect or agriculture aspect is one of the most important, where genome editing technology is applied. And the genome editing technology can be applied to use high degree of genetic variability at a precise locus in the genome. Then potential tool for multiplexed reverse and forward genetic study. It can do precise transgene integration at a specific loci, particularly for the biotic and abiotic trait resistant trait in crop plant, or potential tool for developing virus resistance crop variety. So there are several types of application in the agriculture itself, but particularly we are going to focus mainly on the developing the biotic and abiotic resistant trait in the crop plant. So genome editing has been applied in different crop species. So there are long list. Every day it is growing. So at least 40 different plant species has been utilized to develop different modified genome. And these are a long list. Particularly if you see on the host pathogen interaction part, then whenever any pest or pathogen infects a particular cell, then the PAMP molecule of the pathogen is actually recognized by TRR, and that leads to activation of the MAP kinase pathway and ultimately cause the defense gene activation, which gives a PAMP triggered immunity. But to overcome this PAMP triggered immunity, pathogen also produce different types of effector. Those effector may interact with the R gene or different other host factors, and they inactivate those initial PAMP triggered immunity, cause the effector triggered susceptibility. But when they are interacting with R gene, then they again got captured, and again defense gene activation takes place. So it is a continuous fight between the pathogen and the host. A PAMP triggered immunity, then effector triggered susceptibility, then effector triggered immunity. Like that, it is going on. So if we have to tackle this, then for the genome editing, then we have two different options. Either we can target different plant genes, particularly the susceptible genes, which allow the pathogen to multiply, to grow inside a plant cell. So there are many such genes which allow the pathogen to establish. So we can target those genes for the genome editing, and if those genes are not there, then we can get rid of the pathogen. Or we can also modify the R factors in such a way so that it can activate more to give more resistance. In the other way, we can target the pathogen genes. In one way, we can target the different plant genes. In other way, we can target different pathogen genes, different virulence factor of the pathogens, different gene product of the pathogen, which is essentially required for the pathogen establishment. So if we target those pathogen genes, then also we can get a sustainable management approach. So in these two ways, we can target pathogen or the plant genes, and in that way, we can get rid of the plant pathogen. So similarly, in case of insect also we can do, but insect research is very less regarding the genome editing. So in case of insect, actually majority of the research which are people are conducting, so they are targeting plant gene like in rice, some cytochrome P50 gene has been targeted, which enhance the resistance to hopper and strip stem borer. In case of insect, if we are targeting the insect gene directly, till now we have there is no report that insect gene targeting actually inhibited the insect. Rather, we are utilizing this CRISPR-Cas technology to find out the function of different insect genes. If we want to study the functional genomics of a insect gene, then also we can use this CRISPR-Cas9 technology, and that has been used in different insects. So there is a very latest review in transgenic research in 2021, where this topic has been elaborated in a very elaborate way. Then with respect to the fungal pathogen and the oomycetes, several crop plants, several genes have been identified to target different fungal pathogens and oomycetes, and they found very much effective using this CRISPR-Cas9 technology, and some also use the TALEN technology. Regarding the bacterial pathogens, also people use different susceptible gene like sweet gene of the bacteria or many other genes. So if we target those genes, then it provides the resistance against different bacterial pathogens. And finally, people also targeted different biocontrol agents using different target genes. They activate, they showed the activity of these biocontrol agents and try to find out the mechanism of their biocontrol using this CRISPR-Cas9 technology. So these are a nutshell that this technology in different aspects, starting from the pathogen, fungal pathogen, to bacteria, to insect, everywhere, people are now trying to apply the genome editing to develop a strong management aspect. Now, with respect to the forest pathology, very small work has been carried out. There was a review in 2020. It showed that if you see that this is the total CRISPR publication, and this is the publication in the agriculture and the publication in the plant science, which is going similarly. But in case of forest pathology, it is very less. And even in the forest pathology, maximum emphasis was given in different fruit trees and others, but that the forest tree has not been given that much importance. So the major problem, the lack of the CRISPR-Cas9 research in forest species, is due to three major reasons. Number one, for the forest species plants, very less sequence information are available. Because if you want to develop a kind of CRISPR-Cas system, if we want to find out the spacer sequence, we need to have a large amount of sequences available or deposited in the database. So that is not available for the forest species. Only few forest plant species sequences are available. Then we need to have transformation and regeneration of those forest species, which is almost very few forest species have been known to be transformed, and regeneration protocols are known. And then due to their perennial nature and existence within the semi-natural ecosystem, the regulatory restriction on the genetically engineered trees are far stricter than the agricultural crops. And due to those limitations, actually, the pace of the research work in the forest trees is not that much progressing. But very few woody perennial plants have been successfully engineered with CRISPR-Cas9 system. Majority of them are the agriculturally important tree species like coffee, citrus, and apple. The only forest species with the CRISPR-Cas9 system which has been developed are in the genus of the Populus, and there are different cross and other where people have shown the effectiveness of the CRISPR-Cas9 genome editing. And among this publication, there are only two publications which deals with the pathogen of the poplar tree. So very less amount of research work has been progressed, but there is definitely immense scope in the forest pathology to apply this kind of CRISPR-Cas9 system, because we have very important diseases in case of forest trees like poplar leaf rust, Dutch elm disease, sudden larch death, and white pine blister rust. So these are very much important diseases of forest plants. So people can initiate work on this kind of forest pathology to get rid of the disease using the CRISPR-Cas9 system. Now coming to my own core area, that how we can use CRISPR-Cas9 system in plant virology. So we use this CRISPR-Cas9 system for inhibition of the viral infection, and we can do by two ways. We can target the viral genome as such, the DNA virus and adenovirus genome, or we can target the host gene whose product is necessary for the viral pathogenesis. Now for DNA viruses, if you see, the most important DNA virus in the Indian subcontinent is the Gemini viruses, belong to the family Gemini Viridae. So there are 9 different species, the genus which are under this Gemini Viridae. They have this kind of geminate particle morphology. Then the important diseases they cause are leaf curl, yellow mosaic, and yellow vein mosaic of different crop species. And then they are transmitted, particularly this, I am talking about the bigam virus. They are transmitted by the white fly, a small insect, and they have monopartite or bipartite genome, and sometimes they are also associated with some satellite molecules. So this is the, in nutshell, that what are the important DNA viruses which we work on. And then for the management, we have the insecticide management, utilization of the host gene, then Rnai technology, and the latest addition is the CRISPR-Cas9 based genome editing. So how people actually thought about this? Because just like the bacteriophage infecting the bacteria, when this single-stranded circular DNA viruses infects the host cell, they replicate through a rolling circle mechanism and convert it into a double-stranded DNA molecule. And this double-stranded DNA molecule we can target through the CRISPR-Cas9 approach. So how we can approach? If we express the Cas9 and gRNA from a artificially synthesized Cas9 gRNA complex, that gRNA is actually targeting a particular gene of the Bigom virus, then they will produce the guide RNA and the CRISPR protein, and they form this activated Cas9 complex. And when the viral pathogen infects, then during their replication, the double-stranded phase, this particular active Cas9 complex can bind there and can chop off the viral DNA. And just like the bacteriophage infecting the bacteria, we can also get rid of the double-stranded DNA, the double-stranded form of the single-stranded DNA virus. So there are several reports in last 5-6 years where different Bigom viruses have been targeted by targeting different genes using this CRISPR-Cas9 approach. And in last year, we also targeted the chili leaf curl virus, and where we use different targeting different gene as a multiplex approach. Actually, we take up this chili leaf curl virus, and I'd like to show you what we have got. But before that, I'd like to tell you that how about the RNA viruses? The RNA viruses, for that, the Cas9, normal Cas9 will not work because the Cas9 protein targets the double-stranded DNA. But there are other variants of the Cas proteins like Cas13A or FN-Cas9, which can cut single-stranded RNA. So when a RNA virus infects, they started replication, and during that, if we have a CRISPR and Cas13 component in the cell, which will produce the CRISPR RNA as well as the Cas13 protein, they form such kind of Cas13 CRISPR RNA complex. And when the virus starts replication, they will go and bind to those replicating RNA, single-stranded RNA, and they can chew up. So once the RNA is getting chewed up, host cell do not have any single-stranded RNA repair mechanism. So if the RNA getting chewed up, will not get any virus, replicating virus in the cell, and it will give you the protection. So this is the basic process that how we use single-stranded RNA inhibition by the Cas13A or FN-Cas9. So again, here also several viruses have been targeted using this type of approach. And third approach is that targeting the host gene. Now, in case of single-stranded RNA virus like a Paddy virus, it has been observed that there is a host translation machinery which is utilized by this virus for their own translation. And there is a gene of the virus called a protein called the VPG, or viral genome link protein. This protein actually recruits the host translational machinery, and it interacts with a particular protein of the host, which is called EIF-ISO4E. So if we can inhibit the interaction between EIF-ISO4E and the VPG, then the virus translation will not take place. So we can target this EIF-ISO4E to inhibit the interaction between EIF4E and VPG. And people have used this kind of approach, and using this kind of approach, as people have reported that if we mutate or target the EIF4E or EIF4E-ISO, then we can get rid of different RNA virus infection. Now coming to the last part, that is the case study with the chili leaf curl virus, which we have done. So for that, what we have observed, the key points that earlier people have worked on this kind of CRISPR-Cas9 approach against the Bigom viruses. People have worked with different Bigom virus, Mastri virus, Tapuku virus. They all are DNA viruses, and they use only one gRNA inoculation at one time. And they observed that the gRNA from the intergenic region, which is the origin of replication, that give the best result. But in all this paper, they give rise a concern that after cutting and rejoining of the begovirus genome, many times DNA viruses evade the CRISPR-Cas9 due to escape mutant formation after the non-homologous end joining. So when non-homologous end joining takes place, there is a mutation definitely, but that mutation virus can withstand, and still virus can replicate and virus can come out as a new mutant virus. So that is a real concern. So our hypothesis was, if we target instead of only one gRNA at a time, if we can simultaneously target 2 or more sites of the viral genome through multiplexed gRNA, then we can reduce the escape mutant formation. And to prove this hypothesis, we have chosen experimental system as chili leaf virus and Nicotiana benthamiana as an experimental host plant. So this chili leaf virus, the chili leaf disease actually first reported in India in 1930, but the associated begovirus was characterized long after that during 2006. But after that, there are several begoviruses which has been reported from the chili leaf virus infected plants, though the chili leaf virus was the most predominant one. The disease was become very much severe when there is consecutive 2 epidemics recently occurred in Madhya Pradesh and Maharashtra in 2015 and 16. And after that, it was a real hue and cry for the development of the management aspect of this particular virus. So we developed an infectious construct of this virus to prove that the virus is infective. So we proved their infectivity in the Nicotiana benthamiana as well as the chili plant. And then we start our work. And for that, we have 3 different milestones. The first milestone, as I have told you, the first aspect is identification of the spatial sequence and in vitro cleavage analysis. Then multiplexing of the CRISPR-Cas9 module and implant activity analysis. And then assay for the escape mutant formation after genome editing through T7, U1 assay, and sequencing. So in this begovirus, we choose 3 different regions. Everyone is targeting 2 different genes, and from this region, we choose several NGG motif and several 20 nucleotide sequence. So after initial identification of the different spacer sequence, we further analyzed with different bioinformatics software, and finally we come out with the 6 different spacer sequence, 2 covering from each region. So we are trying to target with different combination in this virus genome so that we can chew up the virus in different portion of the genome. To know whether this in vitro, spacer sequence are really able to clip the viral genome, we did an in vitro assay. For that, we artificially synthesized the CRISPR RNA, procured the tracer RNA to form the gRNA, and then when we add the Cas9 protein, it will form a RNP complex. This RNP complex we incubated with the target viral DNA, and it clipped the target viral DNA into 2 components. So this is the result of this, that this is the viral sequence which we have amplified. And when we allow the different gRNA to cut this viral DNA along with the Cas9, we observe that the viral DNA, which is uncut viral DNA, is cut into different species, different pieces. But our motto was not using one gRNA in combination of different gRNAs. And here you can see when we use more than 1 gRNA, then several fragments, the virus is getting chewed up into different fragments. And that is why we thought that after so many fragmentation inside the plant, if it is happening, then it is very difficult for the virus to again reunite themselves properly in proper orientation to give a mutant virus. Because our aim is to reduce this mutant virus formation and to check the spread of the virus. So then what we have done, we multiplex those CRISPR-Cas9 as a form of construct and then do the implant activity assay. So at the first step, we develop individual gRNA construct using different vector system, which are already well established. Then we join those individual gRNA construct using the Golden Gate technology, and we form a multiplex gRNA construct So we developed 9 different duplex construct and 2 triplex construct. And then we joined those duplex and triplex construct with the Cas9 module, and finally formed a Cas9 multiplex gRNA construct. And then we transformed them into the agrobacterium, and those agro culture was inoculated. The gRNA culture was inoculated at the bottom portion of the leaf, and the virus was inoculated at the top portion of the leaf, so that when the virus starts replication and then goes here, it will encounter with this gRNA, and the genome editing should take place. We determine this genome editing through qPCR and qRT-PCR assay. The qPCR assay of the virus accumulation, we observed that all the treatment combination were significantly lowering the viral titer in this inoculated area. To know that whether there is any significant variation in the Cas protein and the gRNA expression, we also did qRT-PCR of the gRNA and Cas9, where we observed there is no significant variation in the gRNA and Cas9 expression. So at least 3 of the combination actually we observed, they are showing very drastic reduction in the viral load. Now the question is whether, because this is happening in only one area of the plant, so if it is happening only one area of the plant, whether there is any impact on the symptom development in those plants. So what we observed, that in those 3 combinations where we observe the lower viral titer, they have no disease or very little amount of disease, because we are not using any transgenic approach. Only in one leaf we have inoculated, but we are seeing the effect in the whole plant. So this is the result actually I'd like to show. So these are the 3 combinations. You can see in the infected plant, the plant's leaf curl and stunting of the plant. But here with the gRNA treatment, it is same as the mock inoculated plant. This is the second combination, and this is the third combination, where we observe that there is almost no virus symptom and the very reduction in the viral titer. So the question is that whether virus accumulation also reduced in the systemic leaf, because earlier we have done in the inoculated leaf. Now we'd like to test whether in the systemic leaf also virus inoculation lowered down or not. So through rolling circle amplification and qPCR assay, again we have observed that in the systemic leaf also virus titer reduced sufficiently. So virus genome editing causes reduction of the virus accumulation. But our question is whether the NHEJ or non-homologous engineering generated escape mutant formation also reduced or not. So we need to know that whether there is any mutant virus is formed due to this editing and the joining. So for that we need to do assay for the escape mutant formation through T7E1 assay and sequencing. So T7E1 assay is a heteroduplex assay. So if there is a wild DNA and there is a mutant DNA, if you mix them together, allow them to anneal, then some portion of the wild DNA will join with the strand with the mutant DNA. And if there is a hybrid, such kind of heterodimer is formed, then the T7E1 enzyme can cut them and produce such kind of extra band, which will indicate that there is a mutant formation has taken place. But in our result, we observe that there is no mutant formation because there is no extra band which is available here. But in control, it is there, but here it is not there, which indicated that there is no escape mutant formation. So that is our actually target. But to prove this further, we also did sequencing from this area, that whether there is any sequence available with the mutant genome. But after sequence analysis with the TIDE, there is a software, we couldn't able to observe any kind of extra sequence here. So this indicated that there is insignificant mutant formation, and that actually resulted into the reduction of the virus load as well as the symptom expression. So in conclusion, the multiplexing of the gRNA was attempted to inhibit chili-leaf-curl virus infection in Nicotiana benthamiana using Golden Gate and Gateway technique. We developed several constructs, and those among those 11 combinations, at least 3 found very much effective to reduce the symptom as well as the virus load. And T7E1 assay and sequencing revealed that there is a reduction in the escape mutant formation. So this is overall that how we can apply a genome editing tool for management of the diseases. And this entire work, I'm thankful for the Indo-US Getting Overseas Fellowship, DBT, Government of India and IUSSTF. I have done this work with Dr. Hanu Pappu at Washington State University, and also Dr. Michael Neps. They helped me to get all those vectors and other things. And this work I have performed only within 3 months' time. So it is not that you need a very long time or other things. Within very small time, anybody can initiate such kind of genome editing research. So again, I'm thankful to the organizers for giving me an opportunity to speak before you and share my work with you. Thank you. Any question, you please ask me.
Thank you very much. Thank you very much for excellent and meaningful lecture, sir. You emphasize the ABCD of CRISPR to all our participants. Many of us don't know even the expansion of CRISPR, but you given at least some lights on CRISPR and how we can produce the CRISPR-Cas construct and applications in forest, and how we can extend this CRISPR technology in forest research.
Now I invite Dr. A.S. Krishnamurthy, sir. He is Professor Plant Pathology and Registrar TNAU Coimbatore. The technical talk will be on prospects of mushroom cultivation with an emphasis on cooperative Milky Mushroom farming for rural empowerment. Dr. A.S. Krishnamurthy, sir.
Okay, very good morning. This is a good opportunity to meet you all online. Indeed, it's my pleasure to join with the team. Good morning. I take this opportunity to thank Dr. Rajeshwar Rao, Director, Tropical Forest Research Institute, and Head of Forest Protection, Dr. Nanitha Berry, and the organizers and my good friend, Dr. Darshan, Dr. Asia, and then it's my great pleasure indeed. I am very much gifted to be a participant to listen to Dr. Anirban Roy, who has given an exciting lecture. Indeed, I am grateful to you, sir, and all the 80 participants all over India. It's my pleasure to talk to you this morning again. I think I'm going to introduce a little bit technology orientation, and you have been enjoying the science, and now I am taking you for a commercialization of science to technology. Probably you may enjoy. Let me share my slides. Can I share? Hello?
Yes, sir. Yes, sir. Yes, sir. Yes, sir.
Yeah. Hope I am audible now. Right? Am I audible?
Yes, sir. You are audible, sir.
Yeah, good. So friends, it's my pleasure to talk to you on an important topic which can help in enhancing the farmer's income with a simple biotechnology process. So it's rather than science, the technology, how we have taken a simple technology to the farmers will be the next 1 hour talk from my side. Probably all of you appreciate with me mushrooms. They have potential not only for food in many aspects, but Indian vision is a little bit less towards mushroom. Our attempt have been very preliminary. We have not industrialized the technology, whereas it is flourishing in European countries, China, US, and other places. And lots of opportunities are there. Not only for food, they have lots of opportunity. The simple quote by the World Authority, Dr. S.T. Chong, will give you a lot of information. Without leaves, without flowers, and without birds, yet they form fruits. By their attractive color, shape, size, taste, mushrooms have always attracted mankind. And from time immemorial, as a food, as a tonic, and as a medicine, mushrooms are really wonderful creations of nature. Being in forest, being the forest researchers, many a times you might have noticed these kind of fruiting bodies rather than mushrooms available everywhere. Some of them may be below ground and some of them on the soil, and many of them will be on the dead wood. And you might have astonished to see them, and we never thought of utilizing them for the benefit of human being. So my way back, as early in 1988, when I joined as assistant professor in the All India Coordinated Mushroom Improvement Project, I started looking for this kind of fruiting bodies in forests, and everyone, it teaches lots of lessons to us. The fungi in forest, they have very important role, a pivotal role to play in equating the environment, especially carbon footprint. And in such a case, when they are active 24 hours, 7 days, and they are dynamic, when they are growing, they produce a lot of metabolites, maybe an enzyme, maybe a toxin, maybe a growth promoter, and which helps in forest environment in a very big way. Probably all of you may be surprised to know that the largest living organism in the world is not a blue world. It is a fungus. It's a kind of fungus called Armillaria, has been recorded in Pennsylvania State University, covering 2,200 acres with its network of mycelium. Starting from a single spore, it has occupied that much area, and by genetic methods, it has been proved that the entire colony has originated from a single spore. So that way, if you look scientifically, the largest living organism is a fungus. Friends, I have a vision. In India, we made green revolution, of course, white revolution strongly, and a little bit blue revolution. But we never thought of making a strong protein revolution, which is very important. You all, as scientists, accept with me, protein is the base for every living organism, but malnutrition is still there in India. So, to target the malnutrition, if you look to give protein-rich food, none other than mushroom can help you. That's why I designed this flag. At the center, I have put the mushroom cap, and as a food, as a tonic, for the nutraceutical, pharmaceutical, and even for the cosmetic addition, and in forest and agro ecosystem for bioremediation and eco-restoration, these mushroom fungi play an important role. So, we need to give a lot of attention, especially in the forest area, for targeting this fungi for the betterment of human being. So if you look into that way, the best biotechnology industry nowadays looks for pharma products. Biopharma has almost 61% of stakehold, and with Make in India concept introduced by Government of India, we look forward to make our own active ingredient molecules. So far, we depend on European market, or more particularly in China market, for the active ingredient molecules, whereas with the government focus, with scientific knowledge, through public-private partnership, we have lots of opportunity, because Government of India looks for placing India one among the 5 global pharmaceutical hub. And lots of opportunities are there for public-private partnership. In such a condition, the fungi, especially mushroom fungi, which goes with high order basidiomycetes and ascomycetes, they have lots of role play. The expected Indian pharma market by the year 2025 will be around $100 billion. And mostly these products, they look for different portfolio for therapeutic drugs, for cardiovascular, anti-diabetics, antidepressants, very much important nowadays. Everyone is working with stress, so you need to make them free. So antidepressant molecules and anti-cancer drugs. So you all know lots of things are happening because of the COVID pandemic. So all these things can be solved with very good food, especially functional food like mushroom, with lots of nutrition. And by way of introducing this, we can generate millions of job opportunities in the rural area. And it is a scientific industry. Mushroom growing is not just like that. It is an evolved industry. It has started as early in 1600 in Europe, and it has evolved over years. And it is $1 million industry, but it is less understood by the scientists, policymakers, as well as industrialists in this country. So the Pharma Vision 2020 is a big bonanza for the startups. So you look into the pharma growth starting from 2005 to 2020, and it is heading in a big way with millions of dollars looking for young brains, young entrepreneurs, young scientists to join together in a public-private partnership and work for the betterment of Indian farmers. See, a simple example I can give you. Lots of biomolecules are required for these pharma industries to develop new kinds of drugs, especially therapeutic drugs. Shanta Biotechnics, they are trying to make hepatitis B vaccine at the cost of $1.25 when compared to the import value of $125 by other countries. If you start importing from other countries, it will be $125. So in such a way, when things are there, mycotechnology will be the best blend. It's a perfect technology which can answer good health, good environmentalism, and a good business. And we have, with mega biodiversity, especially in tropical forests, with lots of the gene diversity of fungi, we can look for this kind of technology for food and other livelihood, creating millions of jobs. So if you look into that, there are several mushrooms. The top few edible mushrooms, now I am focusing here with button mushroom, many of you might have tasted, followed by shiitake. Shiitake is another interesting mushroom. World over, it is very familiar. And then followed by oyster mushrooms and juice air fungus and Ganoderma. Again, once again, as I told to you, there are a lot of opportunities to go with the different kinds of mushroom. Although certain mushrooms to grow at a very low temperature of 15 to 20 degrees, and the technology is a little bit big. So it is very difficult for a small farm or a medium-sized farm to go with. In the industry, there is other kinds of mushrooms like oyster, Ganoderma. All these things can very well cultivated, and this mushroom loves to grow between 20 to 25 degrees centigrade, and the shiitake loves to grow 15 to 20, and this allows a little bit tropical climate. Ganoderma also allows tropical climate, and apart from all these things, we have another excellent mushroom called Milky Mushroom. Milky, sorry for the inconvenience once again. Okay. Now, as I told to you, although there are several kinds of mushrooms, some of them are industrially oriented and some of them are can be commercially cultivated with the simple technology additions. And we have lots of opportunities to go with the different kinds of mushroom for edible and medicinal. As I told to you, each mushroom has its own climate condition for growing. Some of them grow below 20, some of them between 20 to 30, and some of them love to grow to 30 to 35. So for the tropical climate, we have made a lot of research since 1982 at TNAU, and we have released several varieties of oyster mushroom as well as milky mushroom. Milky mushroom is unique technology. It's our own indigenous technology that I will share with you today. Okay. Okay. See, friends, if you look into the Indian agriculture, as I told to you, lots of crop residue is being generated throughout India in different crops, no? Not only in paddy, wheat, banana, sugarcane, and almost. And even forest, we have lots of woodlots. And these woodlots, sometimes if it is a softwood, for example, if you look into that Shiba pendandra, that is a silk cotton tree, or moringa. So these branches are not having any value. So these kind of softwoods are available everywhere, but we have not used them properly. And actually, if you look into the government target, now increasing the area, and second is to increasing the farmers' income. So to produce more land area, even though it is available, we look forward to climate. So without a proper rainfall pattern, without a uniform distribution, irrigation facility, we cannot expand the land. Whereas in secondary agriculture, we can equally look for producing more profitable produce like mushrooms. So secondary agriculture involves lots of crop residue management. So if you see Delhi, every year the smoke or fogging seems to be a big problem because of the large scale burning of wheat straw in and around that Chandigarh. When you start from Delhi to Chandigarh, on either side the wheat field, so they are burnt. So that smoke causes a lot of trouble. So when we think differently to manage this, and people are doing it in Haryana Agro Industries Corporation to compost all these things in situ, and they are trying to cultivate the mushroom. So similarly, this will help in making manure as well as a profitable produce, mostly edible. And if we look for, we can go for energy industry, even composting technology as is being done in other countries. Even animal feed can be made. So crop residue conversion for food, biomanure, and also pharma, all these things will have a great stakehold in rural stakehold for the betterment of the farmers. So these are all some of the edible oyster mushroom varieties released from Tamil Nadu Agriculture University. And there are false hope among the public that all colored ones are poisonous. Not so. Colored mushrooms are also edible. Even white ones are also poisonous. So a little bit of scientific knowledge and a public-private partnership will help in bringing out so many mushrooms for betterment of the human life. And what is happening in China, they are having more than 50-60 edible kinds of mushroom commercially done, although they are working with more than 2,000 edible fungi. So all these things are very preliminary in India in spite of our repeated research for several years, mainly because the awareness is still lacking among the farmers, farming communities, scientists, and other things. And as I told you, although I can talk on different kinds of mushroom, it is a very big topic. I will just focus on the Milky Mushroom because it's of Indian origin. We released this as early in 1998. So when I started working with this fungus, there were very scanty literature, only 4 or 5 literature available, that too from India. One is from West Bengal, Dr. Purkayasthan Chandra, and another Anila Doshi from Rajasthan, and a few work has been done at IIHR Bangalore. But we started making this fungus commercially viable. So we worked on the cultivation technology, although it looks simple, it's a hybrid technology between oyster mushroom cultivation and the button mushroom cultivation. And we have introduced as a variety in 1998. That is the world's first mushroom variety, especially in Milky Mushroom. Calocybe indica indicates Indian origin. So it's a great treat to the world of mushroom lovers because of so many reasons. Number one is it is tropical mushroom. So it loves hot, humid climate. When I started doing this work as early in 1980s, I felt the mushrooms will come only in cool climate when there is a rainfall and pure dark condition. So all those things, that myth has been solved because of this particular mushroom. So friends, at the time when I was doing this research in Tamil Nadu Agriculture Institute, Coimbatore, I was transferred to a regional research station where the temperature was very heavy. It was a hot summer. I just dumped all the mushroom beds in one of the quarters allotted to me, wherein a lot of seepage was there, and inside the room, the temperature was around 42 degrees centigrade, you may not believe, 42 degree with a high humidity and full light, complete light, because glass windows totally transparent and full light. So under that condition, the mushroom have come up in a big way. Sometimes one single mushroom weighing more than 3 kilo, 4 kilo. So that gave me a lot of confidence to introduce this technology commercially. So it's a great Indian treat, but the story I have written, the story of Milky Mushroom, So that will be more interesting. Sometimes the scientists may get hardship. When you work with that hardships and making the blessing in disguise, you will get a lot of results. And this is an excellent example for that. So in the beginning, we just tried to locate this fungi in the geographical area in and around Coimbatore. And interestingly, many of these mushrooms were found to be associated with the finer roots of coconut. Then we thought the coconut, in and around coconut, we can try to collect the fungus, and then we just cultivated it by tissue culture technology, spawn making, all those things many of you may be knowing. Then slowly we try to make it. Since it allowed excess of cellulose, we try to use whatever possible, like paddy straw or sugarcane bagasse, banana pseudostem waste, everything. And finally, we developed the technology slowly, and that's what I just want to share with you. So the best part, as I told you, is hot, humid climate loving. It can be grown on any kind of cellulosic residue. The shelf life is highly price-worthy. You may not get this kind of shelf life in any of the mushroom in the world. Even for 7 days, 8 days under normal temperature, with a little bit coolness, you can keep the mushroom without rotting. So that is another interesting thing. And then the productivity. Productivity is very excellent in this kind. Now, for example, I will make a small comparison. If you take a button mushroom, with all hardship, you will have to make a compost. And the compost making is a very big industry. It's a tedious process. After making 1 ton of compost, if you are able to grow mushroom, you will be able to get the 300 kg of mushroom. In case of button, even with the best strain, the best agronomic practice, whatever you give, that means 30% biological efficiency. Then oyster mushroom, another interesting mushroom, which can be cultivated with 80 to 100% biological efficiency without composting. That means paddy straw or wheat straw you use, you can directly use it, and from that 1 ton of paddy straw, you will be able to harvest around 800 kg of mushroom normally. So whereas this mushroom, you can harvest, if you are able to use 1 ton of paddy straw without any composting or wheat straw, you may be able to harvest more than 1,400 kg of mushroom. That means 140% biological efficiency, which you cannot get in any of the mushroom in the world. So this is a camel among the tropical mushroom, I can say. So that way. And one thing is, it is having a little bit more fiber. And then the caps type ratio is one of the scientific things that has a very good impact in the consumer market. So if you look into that, if you see any button mushroom, it will have a closed cap. Here also, we are looking for different strains. We try to locate this kind of mushroom, and through our PhD student, we have collected more than 25 isolate from different geographical area. One single mushroom sometimes weighed 13 kilogram. You may not believe it is 13 kilogram. Whereas another similar kind of strain we were able to see with the compact cap having 80 to 100 gram at the maximum. So our target was to have more of small kind of mushroom for the consumer preference. In that way, we started working, and we are continue working on that. And our friend Dr. Anirban Rai has given an excellent example on the CRISPR-Cas technology. Based on the CRISPR-Cas technology, at Pennsylvania State University, they developed a white colored button mushroom with a long shelf life. Here, our target, we are also now connecting with Houston University, USA, and for whole genome sequencing of the fungus. And we look forward to reduce the cap and the stipe ratio, and then fiber content. So more or less, it should look like a big button mushroom. That is our target. And I started in 1980, and now my student is looking on that way to reduce the size in the form of a big button mushroom. Definitely, we will revolutionize the mushroom industry with this fungus very soon. So this is another that isolate that we have collected. If you see this, it is exactly similar to button mushroom. There is no second thought in it. If you cut like this, definitely it can have that kind of value. So this is the mushroom production scenario in TNAU. So many mushrooms are cultivated, button, oyster, milky. Slowly now we are introducing shiitake, ganoderma, all those things. And simply talking, It is again a technology that normally we do. No? We need a spawn. Tissue culture we make from the mushroom fungus, and then spawn is prepared with sorghum or wheat grain under laboratory condition, and that spawn is given to the farmer, and the farmer can use the spawn for commercially cultivating the mushrooms on any kind of substrate, whatever is available to him. So that way here also for Milky also we prepare the tissue culture technology, and then mother spawn, bed spawn, all those similar technology. And then what we do is we go with the paddy straw. Paddy straw is normally available in Tamil Nadu in excess, so farmers prefer paddy straw. Paddy straw is stored in a rain-proof condition, and then it is chopped into small pieces, and then it is boiled or steam sterilized for half an hour to 45 minutes, and then little bit shade dried. And after shade drying, this will be used for mushroom bed preparation. So this is the way, no? It's a small farm in which everything is integrated, starting from storage of paddy straw and then organized chopping, and then it is soaked in water for 4-5 hours, and then the excess water is drained, little bit shade dried, and then steam treated to partially sterilize. No, so major contaminants should be eliminated, insect eggs, nematodes, whatever is available, that's to be destroyed. And normally people also recommend a chemical method of treating the paddy straw, which I do not recommend. So normally they use bavistine and formalin at a 500 ppm rate for treating the paddy straw, but formalin is highly dangerous. I don't recommend that. Normally steaming or boiling is better. No, to enhance industrial activity better, we can go for solarized steaming, solar treatment of these. All those technologies are available, and we can share with anybody, and we can work together in a public-private partnership. Just to share among you the cultivation sequences, I'm just projecting it. The paddy straw is to be steam sterilized and then or boiled, and then it is to be shade dried. No? So under shade, under dust-free condition, it is shade dried until the moisture condition is around 70 to 80%. No? Just to squeeze the paddy straw after preparation with your hand. If you are not having water droplets, then you can think that it is the right stage for the mushroom bed preparation. And we use normally poly bags. And nowadays poly bags are also banned. So we can look for a container system, that is also possible. When you introduce that kind of container system, then semi-mechanization is possible because labor shortage. So most of this handling process should be mechanized. For that also we can do a lot of things collectively, maybe government support, public-private partnership. So that way we can look for different ideas in this container system for cultivation. So normally for a farmer, we recommend a poly bag of 2 feet by 1 feet, and into that poly bag they will fill the processed paddy straw along with the spawn. The spawn is the fungus tissue cultured, and then it is mass multiplied under aseptic condition in the sorghum grain or in the wheat grain. That spawn is taken, and alternating the spawn and the substrate, the bed is prepared in a poly bag of 2 feet by 1 feet, and totally after compact filling, you will have to provide 2 holes for venting, and then it is tied at the top and it is stored. No, it's a farmer who is doing this kind of work. So now, after that, it's for Milky Mushroom, the best preferential temperature is around 30 to 33 degrees centigrade for initial stage. So after preparation of the bed and a dust-free environment in a room, you have to store the beds at 30 to 33 degrees centigrade, and then allow it for 10 to 15 days. Within 10 to 15 days, the entire straw material will be completely ramified with your Milky Mushroom fungus. So he is checking for the complete whiteness. No, once it is complete white, this is the stage at which you have to go for the next step of casing. For oyster mushroom, the casing technology is not there. For button mushroom, casing is practiced. So for button mushroom, composting is a big process, casing is another process, cropping is a third process. Then after processing, then cultivation, processing, all those things. So here we are making oyster mushroom technology for bed preparation, and then button mushroom technology for casing. So once the fungus is grown on the entire bed, now it is time to cut the bed into 2 equal halves. Now full bed is cut into 2 equal halves. Now for preparation of the whole bed, friends, please note, for the preparation of the whole bed, you may need at the maximum 400 gram to 600 gram of dry paddy straw. So dry paddy straw after soaking, boiling, processing, shade drying, and when you fill it into bags, so it may be around 2.25 to 3 kg that bed, total weight of the bed, wet weight of the bed. Now you cut into 2 equal half. You just look in half bed at the maximum we will have 200 to 250 kg or 250 gram of dry paddy straw. So to this, how much mushroom we are going to get, that will be the very surprising fact. So now this gentleman is cutting the bed into 2 equal half. Once the bed is cut, it is little bit compacted, and then after compacting, you will have to apply the casing soil. Casing soil is nothing but block cotton soil or a little bit block loam soil that you can collect any riverbed, near to any riverbed or tank bed. So where you can get that soil, the pH of the soil should be around 8. So this allows more of calcium in the casing. So pH around 8. Black clay loam soil will be better. You need not bother about anything. Collect the soil and remove all the debris. And sometimes if big stones or some clods are there, you just break them. And then little bit you moist them, and then it should be steamed. Maybe in an autoclave or in a steam chamber. You have to steam it for half an hour to 1 hour at 100 to 110 degrees centigrade temperature. This is again to eliminate major saprophytes, contaminants, insects, nematodes, all those things. Maybe after steaming for 1 hour to 1.5 hours, if it is possible, you can go for a double steaming, and then that soil is used as a casing soil. So after the preparation of the bed, after cutting them, you are putting that casing soil. A handful of casing soil is sufficient. Maybe some 500 gram casing soil totally, it is sufficient. After casing, complete leveling, and then the next step is it is taken into a cropping room. The cropping room is very simple that we have designed. Now you can make a lot of IoT based cropping rooms, that is possible, but all these things are our earlier attempts. Now you can totally control everything. Here in this cropping room, you will have enough light passing through over a blue colored silk pollen sheet. Normally the blue light will have an excellent role to play in all the living organisms to create a salubrious environment for growth. So that's what we designed. And then a good aeration is possible through proper ventilation by opening. And you can simply make it. No extra effort is needed. It is half sunken. It is inside. No, it is half sunken in the ground, so that the compactness will help in maintaining the humidity. So temperature is important, humidity is important, light is important, carbon dioxide, air exchange is important. So all these things can be simply regulated in this kind of small chambers. It's useful for a small farmer, but if you want to make a big, again, you can introduce IoT, all possible sensors, and you can have a perfect climate control systems. So this is earlier part that we have introduced in the growing chambers, and you can see a simple structure, and these beds are staked inside, and after casing, you will have to spray water, little bit clean water, potable clean water. You can sometimes, if possible, you can make foggers, misters, everything can be set up. But for a small farmer, he is using little bit of, because when you cover in tropical area a compact cropping room like this with silk pollen sheet, automatically the humidity will be built up inside. Now at the bottom you can spread the soil or sand, and if you moisten the sand or soil, automatically the humidity inside will be around 80 to 85, and on that bed, bed surface after casing you will have to spray. If you make this kind of simple attempt within 1 week, that means first phase is bed preparation 10 to 12 days, and after casing within 1 week you will see the entire bed full of mushroom buttons coming up on the entire surface. So you cannot look every small button to come into a big mushroom, because there will be small reductions. Now whatever is coming in the first stage, you can harvest it within 7 days, and then you allow it for a second plus and third plus, you can continue cropping. So this is the way in which when we introduce this technology, people from other states have come. When Dr. Futela from Haryana Agriculture University, he was the first professor visiting our farmers. You can see all beds full of mushrooms, and they are in different stages. It's getting slow maturity, and then it will slowly grow, and then it is get set ready for harvest. This is again earlier slides. Now our idea, as I said, to reduce the stipe length and to increase the cap. This one should be 1:2. The cap should be big and stipe should be small. And we are looking for the whole genome, functional genome and CRISPR-Cas technology to work with this fungus, and our students are working on it. So you can see robust, milky white and all the beds complete. No? So we can see that. And the best part is, even you can delay the harvest, which is not possible in any of the mushroom, oyster, button, or any mushroom. You cannot delay the harvest. No, it will get rotten. So here, because of the more fiber content, even for 2-3 days, you can keep the bed, keep the mushrooms in the bed. Now, suppose if there is no market demand due to festive occasion or some other strike or some other thing, you can stop harvesting. You can maintain a little bit agronomic conditions, and then you can stop the growth of the mushroom and you can harvest as per your convenience. So, which is not possible in any other mushroom. So, we can delay that. That's what I'm just projecting this through slide. And then it is cleaned, and then it is packed and marketed. So marketing, the best packing technology available nowadays, styrofoam packing, and then using the Areca packages, Areca spath, all those things, even banana pseudostem that can be dried and packing material can be done. So organic green packing is possible. Everything is done nowadays. And then you can see so many farmers, they are trying their level best. Who makes the best crop is the question, and it is full of growth. We have demonstrated to several self-help groups and students. So it is a continuous process since 1998 that we have released this technology. Severally, we have focused, and our All India coordinated project expect all over India. They came, and he was Dr. R.N. Verma. At that time, he was the Director of National Research Center Mushroom, Dr. Jalali from Haryana Agriculture University, Dr. Mukhopadhyaya, Dr. M.N. Kare, so many people. They visited all the farmers, they enjoyed. And then in addition to this cropping, a lot of value-added products we have made, demonstrated over years. And Dr. R.D. Rai from IRI, he was biochemistry HOD, and he also visited the farmers. At that time, he was working at the National Research Center for Mushroom. They all saw and then appreciated the technology, and we have demonstrated. This is a small mushroom farmer, small farmer. So in a salubrious environment, even in forest environment, you can create this even for tribals in a small scale, and we can uplift the livelihood of the farmers. And then we have demonstrated, as I told you, it was the first time in 1998 I exhibited. At that time, the Vice Chancellor is appreciating the technology, and slowly we released it. And then over the years, you can see so many visitors from other countries abroad. So continuously, this is Agri-Intux exhibition. 98 we released, and we exhibited the technology, which gave greater confidence. This mushroom pavilion with a lot of proprietary products that we have developed. And at that time, our Vice Chancellor, he was a cook. He demonstrated how to cook mushrooms. It was a very big gathering. And from 2000 onwards, every year, this Agri-Intux exhibition is coordinated at Coimbatore, and people used to visit and enjoy the treat. And we are also giving technology, sharing the technology to the other countries, even African countries, even Malaysia, Indonesia, Iran. So many people came, they all got that idea training, and our vision is to make big revolution with this tropical milky mushroom. And we are making a lot of other attempts also, and we have empowered rural women to make their own laboratories for preparation of tissue culture, spawn, and then introduce the technology in a big way. Severally. So you can see here, it is practically demonstrated with the farming community. And then there also made them to socially join together in a cooperative way with the NABARD funding support. And you can see here, see jointly they are doing all these things we have done. And then multi-party linkage, creating credit facility, marketing facility through agri-business incubation platform, all those things. And apart from all these things, after cultivation, what to do with the remaining the paddy straw or the spent compost. So here you see, interestingly, among the group, one started preparing all kinds of mushroom recipes like mushroom biryani, and then mushroom samosa, whatnot, everything, mushroom soup, everything. And it was exhibited in a big way in Dindivanam, nearer to Chennai. And all these people did a great job. and continuing their work. And as I told to you, after harvest of the mushroom, the remaining material, which is partially decomposed, was used for vermicomposting, and then vermicompost has been made with the material, and that was also sold. And in addition to that, in some cases, that spent straw, if there is no contamination, it is used for animal feed. So through one single mushroom cultivation, you can go for food, feed, biomanure, enriching the organic content of the soil, and this is possible in a big way. Now, so this is our own technology that is what I just wanted to share with this introduction of mushroom. But of course, to the foresters, I have lots of ideas what to do in forest environment with fungi, and using the other kinds of fungi, even if it is not edible, what can we do? That we can, maybe in coming days, we can share, and a network of knowledge we can create. We can look for joining together to work. It's my challenge. France is for button mushroom, China is for Paddy Straw mushroom, Germany is for oyster mushroom. What is left to India is only Milky Mushroom. And that is a big story. Hope many of the youngsters, we should look for a wider scope with your scientific brain. So as a scientist, be focused, but as a teacher, be wider with your knowledge. And that is what, who is brave enough to think, bold enough to believe, and talented enough to do, will achieve whatever they wanted in life. This is a lesson from Apple, and we tried, and from scratch to the technology development, TNAU has made very good attempt this world-class technology in variety, and we have released it as early in 1998, which has been appreciated by Dr. S.T. Chong, who is the world authority, a professor who is responsible for completely revolutionizing mushroom industry in China. Now, even though mushroom cultivation started in Europe, grown in US, now the Chinese are the leaders, even in all mushroom, butter, oyster, shiraki, medicinal mushroom, mushroom-based preparations, product, everything Chinese are dominating. I had an occasion to meet Dr. S.T. Chung 2-3 times in international conferences, and I also attended one medicinal mushroom conference at China. They are doing a lot of work, and similar possibilities are with us. We can look for lots of opportunities for food, feed, biomanure, pharma industry, why not agrochemical industry, through mushroom and mushroom fungi. So my final statement to you is, even the termites have their own fungal garden. You might have asked the termitorium. Termitorium keeps the particular fungus. The termites are talented enough to harvest that particular mycelium alone, and then Termitomyces robustus. There are so gregarious, so many species are there. The termites are clever enough to collect that fungus alone and store them in their fungal garden to enrich their young ones with nutritious feed. So that fungus mycelium that contains an excellent immunomodulatory protein as well as an excellent sugar. Trehalose is the sugar available into that. For that particular purpose, the termites collect them and store them in the fungal garden, but the fungus is clever enough, they grow the remnants, termites remnants in the hive, and then they come out as a mushroom. And in that soil, if you collect the termite soil, you can get a lot of actinobacteria. So all these relationships are going on. If you are able to look very strongly into that, we can harvest a lot of bioinoculants, biocontrol agents, growth promoters, everything. Similarly, in the forest ecosystem, I used to astonish with the leafcutter bee. Leafcutter bee is an excellent one that is normally available, and also leafcutter ant. Wherever the ant movement is there, definitely you can look for a fungus called Leucoagaricus gangliophorus. The ganglion of the fungus is filled with a lot of the honey sugar, that is trehalose. And these ants are clever enough to collect that honey sugar and feed their young ones. And this honey sugar, as well as the immunomodulatory protein, has an excellent role play in growth promotion as well as health benefit. So if you are able to look into that, we can make a lot of activities in forest area for the betterment of livelihood of the farmer, and also scientific additions for making a lot of patentable molecules. So that is my vision. Hope to join with you in some other session to talk more on fungal biomolecules and their value for industrialization. Thanks for the opportunity given to me. Jai Hind. May God bless you all. Thank you very much. I am ready to answer for your queries.
Thank you.
Thank you so much. Thank you so much, sir, for your excellent presentation. And we are very much interested to collaborate some with you in near future. I am doing agroforestry also, sir. So I saw one of the drone picture that is you are using with the coconut trees and mushroom cultivation. So we are very much interested to involve this in as a multi-tier cropping system also, sir. And it's excellent, sir. First time I saw this Milky Mushroom, sir.
Thank you, madam. Thank you. Thank you very much. And we can look for TNA is looking for any kind of collaboration. And Dr. Darshan is our student. He knows well what is TNAU strength. And we have lots of facilities to do any kind of ready for any kind of collaboration. Darshan is there. He knows the strength. We have a lot of equipments, facilities to do basic research, to applied research.
Sir, yeah, definitely we will contact you in near future also, sir. One more thing, sir. I have one question, sir. Please, can we replace this Paddy Straw with cocoa coir, sir?
Coir waste, no, it is full of lignin. No, for that can be used for cultivation of lignicolous mushroom, like Pyrotus or lentinus, so like that, shiitake, all those things. Even woodlogs can be used, but this milky mushroom allows more of cellulose. So more cellulose addition would be better.
Okay.
How this mushroom can be included in agroforestry model, sir?
Yeah, it is possible. Wherever you have that agroforestry model, the dried branches can be collected, and the dried branches can be processed, and through agar, we can introduce the fungus, whatever you feel like. For that sake, I feel it would be better to go with the auricularia. Auricularia juice hair fungus is another excellent mushroom. It has a very good international market, and it has a very because of its fiber content, vitamins and calcium addition, iron content. So it has very good potential that we can definitely do it. It is possible. And also shiitake. But all the shiitake loves a little bit cool climate. Maybe wherever you have that kind of system, we can help you and we can definitely do that. Ganoderma is another potential fungus. Although it is known as a disease-causing fungus, it is medicinally highly valuable. More than 200 proprietary products, including Ganoderma soap, Ganoderma soup, Ganoderma tablet, Ganoderma decoction, Ganoderma chocolate, Ganoderma oil, everything, Ganoderma, wine, everything is available internationally. Why not we? And of late, one of my students has done an excellent job using the Ganoderma extract. We have identified an active biomolecule called squalene. Squalene, with its potential, could help in controlling the virus diseases, especially groundnut bud necrosis virus. And of late, we have published 2 excellent papers, and that speaks better on the value of the metabolites of this kind of fungi in the management of potential diseases, both viral, bacterial, fungal. And I can also share another interesting mushroom called the Chinese caterpillar fungus. Ophiocordyceps sinensis, Ophiocordyceps militaris, then Ophiocordyceps neovalkiana. All those things have potential species. Then a lot of puffballs. So all these things will have an excellent role in agroforestry ecosystem. And some of them are mycorrhizal even. They will help in my afforestation programs also. So lots of I do work in forest area in horticulture college and the forestry college in the horticulture research station at Ooty, and I have surveyed so many forests. And then at forestry college also we tried, and wherever you have that eucalyptus plantation, you will get a Physolithus. Physolithus and eucalyptus has an excellent role play, and this Physolithus accumulates golden nanoparticles. So nanotechnology additions is also possible with this kind of fungi. So all those things we can have very good discussion maybe in coming days. Thank you very much.
Nice lecture, sir. This is AJK Asaya. I would like to ask some questions from chat box. Mr. Venkatesh has asked one question. Can mushroom cultivate?
Not audible. Hello.
Huh.
Can you,
Sir, sir,
yeah.
question is, can mushroom cultivated under bamboo?
Yeah, there is one particular mushroom called Hericium erinaceus. It is called bamboo sprout, and that can be introduced in the bamboo plantation as a mycorrhizal, and that can be very well used. So that is having excellent molecules that can enhance your neurons. And one of my good friend, Dr. Vigneshwari Shabaratnam, working in Malaysia with a medical university. Both of us are trying to find out lots of opportunities for that Hericium erinaceus biomolecules. And lots of literatures are also available. So if you are interested, we can definitely share the technology.
Thank you, sir. I hope, Venkatesh, you have got your answer. Sir, I want to ask one question, sir, personally.
Please.
What would be the temperature in that harvesting room, sir?
Harvesting room for which mushroom? Milky mushroom?
Milky mushroom, sir. Milky mushroom.
Around
What
30
you
to 35. It is a tropical kind of mushroom. So you might have seen that blue shield pollen sheet made that cropping room where you will get to full humidity, high temperature, 30 to 35 with more sunlight.
Okay, sir.
Yeah.
Okay. Sir, what is the cost, market cost, sir, and market response cost?
Almost for all these mushrooms, the production cost little bit varies. No? From oyster, milky, button, the production cost may vary from 80 rupees to 100 to 120 rupees, depending upon the mushroom that you are cultivating per kg production cost. Whereas in the market, it is being sold for 300 to 400 rupees normally. 200 gram pack around 40 rupees. Just imagine. 200, 250, 400, sometimes it reaches 400 even. So that depends upon the market.
Thank you very much, sir. I feel Darshan is very fortunate enough to have you as a teacher.
Thank you very much. I'm blessed to have Darshan also. Darshan is a hard worker, very good scientist. DNA is very proud of such kind of alumni. And Golden Jubilee, we are celebrating Golden Jubilee at TNAU. I welcome all the scientists to celebrate with us whenever you feel like. Please come to TNAU. It's a welcome from my side.
Yes, sir. I am always grateful to be part of TNAU. Yes. So really, I am blessed to teacher like you, sir.
Thank you.
And now I request Mr. Asaya, sir, to give vote of thanks. Thank you, sir.
Thank you.
Good afternoon to all. Both the sessions, I hope it was very good, and thank you for giving me opportunity to deliver vote of thanks. First, I feel honored and privileged to thank Professor Dr. A.S. Krishnamurthy, sir, Registrar TNAU, for your informative, thought-provoking talk, sir. We are thankful to you, sir. Despite your busy schedule, you honored us with your presence. My heartfelt thanks to Dr. Anirban Roy, sir, Principal Scientist, IARI, New Delhi. Your informative talk, which will definitely help us for doing further research in management of insect pests and diseases using molecular tools. Thank you for honoring us with your presence. My sincere thanks to our director, sir, Dr. G. Rajeshwar Rao, sir, and our group coordinator research, Dr. Kundu ma'am, for their constant encouragement and support to make this webinar possible. I am thankful to my HOD, Dr. Nanita Berry, who works hard to make this webinar success. My sincere thanks to our report chair, Mrs. Shalini Bhote. Thank you, Dr. Darshan. Dr. Darshan is very energetic, intelligent scientist for excellently conceptualized and conducting this webinar. Our thanks are also due to all the participants, HODs, scientists, technical cadre officers, staff, colleagues, and all online audiences who find time to attend this webinar. I am personally thankful to all the staff of Forest Protection and IT section, whom constant support we got for organizing this webinar. Last but not least, a big thank you to one. Now, with the permission of Director, sir, I declare end of this webinar. Please do visit TFRI whenever possible. Thank you very much. Thank you, sir.
Thank you all. Thank you very much.
Thank you.